So we're going to talk about protein folding and what that means to Snowflake, or why we are talking about it from Snowflake. So quickly run through the agenda. We're going to explain the business context because we understand not everybody is as passionate about life sciences probably as I am. So we'll break down what that means and why it's a big deal. We'll talk about what we can do with that in Snowflake. We do stuff with different capabilities that were announced in the last days and the last months. So it's a good way to see how we can push the boundaries of Snowflake in an innovative industry area. We'll have a demo that shows how these capabilities come to life. And finally, we'll see whether outcomes really mean something. It's not smoke and mirrors. It's actually grounded in reality. So I'm the field CTO for life sciences. So what that means is day in, day out, I talk to life science customers to make Snowflake relevant in the context of pharma industry. My background has also been that. I've been a bioinformatician in the past. So I breathed this for most of my life. So with that being said, the Gen AI in life sciences is beyond chatbots. So there are quite a few exciting use cases. You look at the top, we have the whole R&D that focuses on very domain-specific models that are based on transformers and LLMs in the industry. So today we're going to focus on one particular area, which is called drug discovery models, and we're looking at protein structure prediction and chemical property prediction. That's the aim of what this demo is. But if you ever wonder where in the industry it fits in the R&D part of the value chain. Why is it a big deal? So just for context, it's a 1-minute summary. If you haven't done biology in your life, consider it a recap in 5 seconds. Everything that is regulated in the body, whether you have cancer, don't have cancer, you're normally functioning, depends on that thing that's floating. That's called a protein. It has a specific structure. It's encoded by things called amino acids. And having the structure defines whether it's functioning well or not. So life sciences spend millions of dollars trying to figure out what is the right protein to target for a disease. And that's done by many different techniques, including something called X-ray crystallography that used to take months to figure out what is the shape of a protein so that they can define a drug to work with it. How is AI being used? Of late, because of AI and generative AI, people can actually de novo find the structure of this protein by looking at just sequences. So you don't spend months in the lab, but you feed the sequence to a custom transformer model, and you actually get what a structure should look like. And this is real. There are companies that are today making money out of what we call AI-based drug discovery. There are at least 4 or 5 drugs in the clinical trials that have been discovered through AI one way or the other. And the group that does it, or the companies that would do it, you will see within target identification, or you will see it as tech bio. Just a quick recap of how much of money we are talking about. There's like billions of dollar collaboration for tech companies to work with pharma to find the next drug using AI. So it's not a 100K, 200K POC, but these are real life use cases which have millions of dollars investment behind, because you're essentially accelerating how things were done in the lab before. Very recently, if you follow the news, DeepMind, one of the Google ventures, actually launched AlphaFold 3, which is another path-breaking generative AI solution within the fold of protein folding. And go read up the paper. It's very interesting on how they leveraged it and how it's being used in the industry. So recap, right? Gen AI is accelerating understanding. We can predict structures. It's not just predicting content. You look at a sequence, you get a structure that's established, and some of the models are like what we call AlphaFold. Prior to Gen AI, took months. Now people can do it in minutes or seconds. I wouldn't say minutes, probably a few hours, depending on the size of the protein, but it happens, and there's a lot of compute involved. And the groups that does it today are called customers in R&D IT, computational biologists that actually work on this problem as we speak today. If you were at NVIDIA GTC, a lot of time was spent with companies that actually brought their high-powered computes like H100s or Teslas to power this kind of work. Bringing it back to what's in Snowflake. We launched a lot of capabilities within generative AI, right? Cortex was talked about last 2 days. If you follow stuff, Cortex was spoke about a lot. So we have many abilities to where we use Gen AI. We can bring our own models, or we can use the models that's hosted within Snowflake natively. We can also do other stuff, like we can store all of them as vector data types and do similarity search and then invoke the model on demand. So for the purpose of this demonstration, what we have done is leveraged a combination of these. We built our front-end facing app in Streamlit, so that's the web app experience. We stored each protein using a custom transformer model that's called Protify, and we hosted that in our container services. Because remember, these large language models are fine-tuned on millions of proteins, so you need them to be domain-specific models. You can't use a Mistral or a Llama or general text-based models. So those specific models that we talk about, we can bring them into container service. That's what we did. We then embedded a knowledge base of 500,000 proteins that we could already find in the public database, converted them to dense embeddings, and stored them in a vector data type. And then we ran a Cortex similarity search, found matching proteins, and then display them back in Streamlit for an end user. So the end user goes from a sequence to all these behind-the-scenes process and gets structures back. In addition to confirm if the structures are real, we also called NVIDIA, a partner, and their particular drug discovery model called BioNeMo to get them to also fold it using a large language model they host. And you can compare if all the three structures match or not. You will make more sense when you actually see the demo. So with that, I'm just going to bring up the architecture. We went through this. This is what it all flows into. So the sequence itself is a bunch of alphabets, what we call amino acids. It resides in internal managed stage of Snowflake, which is our S3 here. We hosted the Protify on container service, so it encodes each sequence into a dense vector embedding. We throw that into our vector data store, or it's a native vector data type. We then invoke the similarity function, the cosine similarity, the L2 distance here, and then we find the top 3 hits that match this particular sequence that we loaded in. And then we display all of that, and you can see if the proteins are structurally similar, and we also call a metadata to service to see if they are functionally similar. So it's a nice use case of bringing 3 different modalities of Gen AI in action. We use containers, we use Cortex, and we also call an external function. So it's a cool way to see how you build an app. The vector embeddings look like this. Each of them is an identifier followed by a dense embedding of the protein. And the output, the 3D structure you see, what you get back is what we call a coordinate file called PDB file that also is stored into a Snowflake table. All of this is done behind the scenes. When Jason runs a Streamlit, this is what happens. But I hand over Jason to take it forward from here.
So we use Proti5 model. This is an open source model to create custom embeddings. So Proti5 is a transformer-based model, pre-trained a large corpus of protein sequence data to generate inputs and labels from those protein sequences. As such, this model can be used to create embeddings on arbitrary protein sequences or amino acids. So here is a feature extraction overview on how Proti5 model can be used to derive embeddings from arbitrary protein sequences and how they can be used for downstream classification tasks, for example. So let's say our input sequence is SEQ in this case, and then we tokenize our input sequence and also add positional encodings on top of that, and then we pass the resulting vectors into the transformer model, and then do the pooling to create fixed embeddings for our protein sequences. And with that embeddings, we can use Cortex vector similarity search to identify similar proteins. So the specific model we use actually only contains the encoder portion of original Proti5 model. The benefit is that it's easy to host and performant, easy to host in Snowpark Container Services. And also, it's very lightweight as well. So you get the cost benefit in terms of the model that you're hosting. With that, I am going to show a demo of these Streamlit apps. So here is a Streamlit app that we built in container service. And then our input would be a protein sequence. So let me set this correctly. After we input the protein sequence, we're doing the live query of essentially the Snowflake database and then rendering the 3D structures of the proteins. So for example, our input sequence is here, and then it's a tumor suppressor is our input protein. And then we are able to identify 3 similar proteins that are also tumor suppressors. So the key here is that, right, just from observing the structure of the proteins, you understand essentially the functions of the protein, and that helps a lot of researchers to accelerate the drug discovery process. And we can try even, for example, different protein sequence now, and we'll be able to get live updates based on your Snowflake data sets. All right, so these three are also all toxin as well, and Snowflake and Harini will talk a bit more about the results. So here is a Streamlit demo, and what Snowflake, what the data stored in Snowflake looks like. It really just looks like, for example, we have a Uniprot ID, we have our sequence, our protein sequence data, and then we also have the embeddings that we created with Protify model, as well as essentially a short descriptions of the functions of these proteins. And then here is where we're using the Cortex vector similarity search. So we're using vector L2 distance, and we're passing our input protein sequence, and we limit 4. We order by distance. So the top 3 would be the most similar proteins given an input of the protein. I do want to do a quick code walkthrough as well, just to show how easy it is to do in Snowflake, in Streamlit, to build a solution like this, right? So at a high level, you can either build your Streamlit app in a container, Snowpark Container Services, or Streamlit in Snowflake as well. So what I'm showing here is a Streamlit in Container Services. So we use these environmental variables to credentialize into your Snowflake account, and then we set up a session so that it builds a connection of your Streamlit app in container service to your Snowflake account, where you can define your database, your schema, even the warehouse as well. So with that, we set up a session, and then we create some, essentially these are just some functions that I can visualize my PDB file, my protein data bank file created by NVIDIA BioNeMo framework, and then as well as, I guess, some functions, really essentially just my SQL command based on the passing protein sequence. And then here is where actually the Streamlit code happens. Very easy, very simple. It's only 30 lines of code that I'm doing both visualization of the protein structures as well as doing the Cortex vector similarity search to identify the similar proteins. So really, this SQL query is where the secret, really kind of the Cortex piece comes in. The vector L2 distance search given an input protein sequence. All right, with that, I'm going to pass back to Harini to talk about the results and next steps.
Cool. Yeah, thanks. I think you saw the demo, but just to summarize, right? We passed the sequence. So the normal world, we don't know what it looks like. And what we found the match were exactly, as you can see, two proteins here, at least, and the 3 that is on Streamlit, are the exact same function identified in blue in the slide. Same thing with another sequence. And what I've now done in this slide is blown up that into the 3D structure. And you can see that structures look similar. And what we started was just a text, just with a few hosting a transformer, doing a similarity search. You're actually able to get something that's very, very difficult, which used to take months in the lab. So it's pretty amazing if you have ever worked in that area. Same thing with another sequence, right? You saw tumor suppressor. Tumor suppressor is a protein that is normally associated with cancer. Most of the drugs you develop for cancer try to find this as your target. Same thing. We gave a sequence, and then we found that you found exact same matching functional sequences just by doing a combination of different functions within Snowflake. What next, right? Where do we take it from here? Or why Snowflake? That's the first question we get asked when I present this to customers in life science. First of all, it's about security and governance. You can do this everywhere, I guess, now if you have an ability to call and hosted LLM or an inference endpoint. But the idea is we don't want the data to move. So every time you bring a sequence store and manage it within Snowflake, you can now do the whole value proposition, including searching, folding, and displaying back in Streamlit in a secure environment without having to do external calls here and there. Second one is, within R&D, it's a big function. We start with proteins, but there are other functions that are part of the value chain. We want to extend that across the value chain. So you start with a large molecule, and you want to go slowly into chemistry. You want to go into more preclinical. So this is a good way to integrate everything into a centralized lake. Third one, very important, what we started Snowflake with is data sharing. All of this data is terabytes in size. And most of the time, pharma work with collaborators. They don't work alone. So this is an excellent opportunity for people to share their sequence data or the research collaboration into a Snowflake environment and not have to worry about having to do the analysis separately outside. So this just brings more compute to data with having the data shared from say, an academic research center, for example. And last one, The ability for warm compute pools cannot be understated with container service. Hosting something like Protify is not probably difficult, but if you want to bring a bigger transformer model, and that needs to be readily available every time you want to call a sequence, convert that to an embedding, having a warm pool is extremely effective, and I think container services helps that story. And then having Cortex integrated with it for then optimizing it and using managed LLMs for other tasks is also another great chaining of what we call these LLM agents. Next steps, we would love to work with NVIDIA more to see if we can host some of their other drug discovery models. Discussions in progress. Probably move to more advanced generative AI, like stable diffusion models. We have something in drug discovery called DiffDoc. That's pretty exciting. We'd love to see that. We'd love to replicate the same in the world of small molecules. Something our customers ask in life sciences, can we find similar compounds all the time? So looking to see what we can do there. That's it. Hope this at least gave you a peek of what we do with Snowflake in the industry and got you excited in the space. All right, thanks
Good.
everyone.
Thank you.